Hello and welcome from live from the American Academy of Ophthalmology meeting in New Orleans, Louisiana. It's great to be here in person, and it's great to be coming to you live from the American Academy of Ophthalmology. I think behind me you can see all the hustle and bustle that's still here, and all the colleagues who are reconnecting, and all the work that's being done and the information being communicated. Pleased to be joined live today by Dr. Mitch Brigell, who, due to a last minute scheduling change, couldn't be here in person, but has still graciously agreed to participate. Dr. Brigell has spent a large portion of his career in the pharmaceutical industry and translational medicine, and helping to bring therapeutics to market that have made a marked difference in improving the sight and preserving the sight of tens of thousands of people around the world. He has extensive experience in both bringing those products to market as well as doing the clinical trial verification and validation of those. And I'm delighted to have Dr. Brigell join us today as we broadcast live, although he, as you can see, is not live here. It's in New Orleans, but is live from another location. So welcome, Dr. Brigell.
Thanks, Jim. My pleasure to be here. Sorry I couldn't be there with you.
Oh, so are we. But we're glad to have you here with us through electronic medium. So one of the things we wanted to talk about today was just bringing those therapeutic drugs to market and the clinical trial, and the role of electrophysiology in both the safety as well as the efficacy of new drugs that are proposed to be introduced and taken to the FDA for their approval. So, Dr. Brigell, can you give us a perspective of that?
Yeah, well, as you know, there are many stages of drug development, and in many of them, the ERG can be important, both, as you say, for safety and for efficacy. In early drug development, it's really important to get some sign of efficacy as early as possible. And oftentimes, the clinically, quality of life and the clinically meaningful visual changes that one sees with therapies take, years, months and years to develop. And you don't want to do a trial for 3 years to show that your visual field's improved in glaucoma in order to prove concept. You basically want some kind of measure that can tell you your drug is having a positive effect on the retina as early as possible. And the ERG is able to do that. In terms of making decisions about whether to bring a drug forward, into phase 3 very expensive trials, a measure like improving the physiologic health of the retina can be instrumental in saying, yes, if we do this 3-year pivotal trial, the drug will have a good likelihood of succeeding. So it can give you an early sign of efficacy that can move your program forward. In terms of inherited diseases, clearly the ERG is well accepted as an endpoint. I'm not sure that FDA would accept improvement in the ERG as an endpoint, but certainly for decision making, as I said, it can facilitate decision making. In terms of safety, the ERG also can be very important. And in preclinical safety studies, the toxicology studies, the FDA is requiring more and more drugs to show that the ERG is not affected adversely by the drug therapy. So, if the drug is affected adversely by drug therapy in an animal model, it in and of itself doesn't halt development of the drug, but it means that you'll have to monitor humans to see, is there an early sign of an adverse effect on the ERG in humans. So ERGs are often used as a safety measure to be sure that there's not an adverse effect on the retina. In many cases, an ERG effect can be reversible if you withdraw the drug. So for the safety of the patient, you see an ERG finding, and before there's any atrophy or any permanent damage to the visual system, you can stop the drug. So it can be critical in helping assure the patient and the agency, the FDA or regulatory agency, that your drug can go into clinical trials safely. And so it can change the whole balance of the risk benefit. Finally, many drugs, once they're on the market, people start complaining of visual symptoms, and the FDA could say, well, we want to understand this better, and that could mean instituting a trial using ERG to show that the visual symptoms, are they related to damage to the retina or not?
So, traditionally, a lot of people who are ophthalmologists and maybe even scientists such as yourself, when they think about electrophysiology and ERGs, they think about this massive piece of equipment about the size of a desk,
That's right.
very expensive to come in, having to dilate eyes, put an electrode into the eye, have someone specialized conduct the test, and then you get the waveforms out and have someone who is a professional in interpretation of the results. And one of the things that you did was in a trial was implemented that utilizing this handheld device that we make called the Retival device. And so perhaps you can talk for a few minutes about why you chose to implement this device in your trial and how it's changed the utilization of ERGs in clinical trials.
Yeah, good point, Jim. I think the knee-jerk reaction you get from a lot of clinicians when you tell them you're going to put ERGs in your protocol is don't do it because of the history of the patients not liking having their pupils dilated, having to put a contact lens in, having to sit in a dark room for 20 minutes to dilate. To dark adapt, excuse me. The Retival has really changed things dramatically. Number one, because of its portability, it's easy to have in the clinic. You don't need a dedicated room. The skin electrodes and the engineering to modulate the light depending on pupil size and not need dilation make the test really very tolerable for patients. Patients don't mind it in general. It's definitely much easier than doing a visual field for the patient. The other thing is the kind of turnkey aspects of the Retival. I mean, like you said, there doesn't have to be a whole lot of training to do the test. We had 20 clinics use this device, none of whom had ever done ERGs before, and we got great data, very repeatable and very equivalent between centers with a minimal amount of training. Basically, they watched the video, answered a few questions, and submitted some data to LKC to show that they could do the test properly and place the electrodes properly. So it was easy for the site, it was easy for the patients, and we got great data. The device can be programmed to whatever tests you want. So if you're doing a glaucoma trial and you just want to do the photopic negative response, you can just have a protocol that says photopic negative response, have all the parameters you want in it. The technician just has to dial that protocol and place the device over the eye, and it takes care of itself. if you're, we were doing a diabetic retinopathy study. We were looking for an early sign of efficacy. And for that, the flicker, the standard flicker screening program that is on the device, we just used it. And it was very easy for the site, very easy for the patient. And we got very useful information from that. And if you're doing safety, you can do, if the FDA is requesting in ICEV standard protocol, this device, complies with the ICEV standard, and it can be done. There you have to dark adapt for 20 minutes, but you don't need a special room. You don't need to place electrodes. It can be done as non-invasively as possible to satisfy safety requirements from the FDA.
And what about integrating with the protocol that you implemented for your diabetic retinopathy trial and actually transmitting the data and get that integrated so that it was all together?
Yeah. Yeah. Well, we worked with LKC to see the parameters that we wanted. LKC maintained their own database, at least for one of the trials, and they did a transfer agreement with our CRO. They transferred the data, and we had a very secure way of integrating the data on a monthly basis with the clinical database. And then once the data was in the clinical database, our data management and statisticians were able to treat it just like any other laboratory data or clinical data that we obtained. So it was quite seamless. In another trial, we used a third party to maintain the database, but in that case, we had an expert reader review the ERG, make sure that all the cursors were placed correctly, that the responses were valid, and then to make any edits that were necessary, and then transmit the data to the database that was maintained by a third party. So in both cases, it really worked very well, and we were able to get very quick turnaround times on the reads, and we didn't have to wait a long time to analyze the ERG data. It was ready for our top line results.
And for someone who's getting ready to begin a clinical trial such as this, what advice would you have them in terms of cost effectiveness, in terms of them wondering whether or not a device that's this size versus the traditional ERG in terms of quality of output and and availability and so forth? What would you say to those folks who are considering?
Yeah, I mean, the traditional ERG is not something that's friendly to clinical trials. There's variability in equipment, between sites. This device, you're able to provide to all sites for really, reasonable amount of money. We've worked out, rental agreements with options to buy for our sites with LKC that have been reasonable for the company. Plus, once the sites actually learn the value of the equipment, they can buy it and have it. And what it does is standardize the test across sites, which is very difficult to do in a standard ERG. These devices are very well calibrated, and you get very consistent data from one site to the other. You don't have to worry about differences in the brightness of the flash or how the filters are set. You set the protocol on this device, send it out to 20 sites, use this protocol, and you're going to get consistent data across multi centers. that's not easy to achieve using standard equipment.
All right. Well, I appreciate your time today, Dr. Brigell, and appreciate your perspective on utilization of both electrophysiology and ERGs in the clinical trial world, and also your perspective on standardization and using a device such as the Retival, which is a very portable device that's easily implemented across many, many sites. So I thank you for your time. It's great to be here live in New Orleans at the American Academy of Ophthalmology once again. Mitch Brigell, I'm very sad that you're not here in person, but I understand the scheduling change that you had, and wish you every success in the future. And thank you very, very much for your time and your perspective.
Yep, I hope to be with you next year at AAO. Thanks, Jim.
We hope to see you here as well. Have a great rest of your day, and thank you all for joining us today from New Orleans, Louisiana.